All right, hello everyone. So my name is Sam Newtile. I'm a professor at Penn State Barrett in biology. And I'm going to be presenting some work that I did with two of my colleagues, Dr. Adam Simpson, who's also in biology at Barron, and Amanda Harwood, who is a professor at Alma College in Michigan, looking at remediation of a DDT Superfund site using activated carbon. So as I'm sure all of you are, or most of you are probably aware, DDT was an insecticide that was used pretty heavily in the United States between the 1940s and about the end of the 1960s. This compound was originally discovered, or its insecticidal properties were originally discovered in the late 1930s, and it was used pretty heavily during World War II abroad in the Allied war efforts to control pest insects like mosquitoes. The efficacy of this compound overseas led to its usage within the United States to control similar pest populations like mosquitoes, and it proved to be kind of a miracle chemical. So these are some pictures of the kind of indiscriminate widespread use of DDT that occurred between like late 1940s, early 1960s within the United States. So we can see here that there was really no concern over potential toxicity of this compound to humans or the environment. So it was sprayed in large quantities in public spaces. You can see that poor kids getting blasted in the face with it. They were spraying these kids in pools. They're shooting it on beaches, and then there's even advertisements for using DDT in the home where food is stored. So there were no really acute effects of DDT on humans, but over time we kind of learned through this indiscriminate use that DDT does have some nasty side effects, particularly in the environment. So DDT, being a hydrophobic organic compound, tends to accumulate within soils and sediments of wild places. The hydrophobic compound will stick to organic matter in soil and sediment, and from there can accumulate within invertebrates that spend their time within these sections of the ecosystem. In aquatic ecosystems, we see that DDT will make its way from sediment into invertebrate species that are then fed on by fish that can then be predated upon by other organisms such as birds of prey, raptor species. The transfer of DDT up the food chain is what we refer to as trophic transfer, and due to the widespread indiscriminate use of this compound, we saw that DDT within these food webs would actually magnify as it moved up the food chain. So as we went from invertebrates to our large order predators here, we see that the DDT concentration accumulates steadily through that food chain. The accumulation of DDT within these top predators led to extensive declines in bird populations within the United States, most notably the bald eagle. So bald eagle population significantly declined between the 1940s and the late 1960s, and it was eventually linked to this uses of DDT. So within birds, particularly these raptors here, we see that DDT causes eggshell thinning. So the DDT that accumulated within the tissues of these birds would be transferred into the eggs from the females, which would cause changes in the way that calcium was deposited into these eggshells, such that they became thin enough that when the birds would incubate them, they would step on them and crush them, killing the offspring before they had a chance to hatch. Killing off the offspring caused those large declines in the bird populations, which we saw across the United States. So eventually we kind of put two and two together that DDT was the cause of these bird declines, and in the 1970s with the formation of the EPA, we finally saw a United States ban on the use of DDT. This banning, however, of DDT in the 1970s was good because it stopped the kind of indiscriminate use of this chemical, but we still see ramifications of this indiscriminate use today, almost 50 years later. There are sites across the United States that are still heavily contaminated with DDT at concentrations that are just exceptionally high. And one of those places is the Velsico Chemical Corporation Superfund site located in St. Louis, Michigan. So this location is located along a river known as the Pine River near St. Louis, Michigan. And there was a chemical plant, the Velsical Chemical Corporation had a manufacturing plant there that was in operation between 1936 and 1977. During this time, they produced many chemicals, including DDT, and through some poor business practices and improper waste management, the operation of this plant led to some pretty extensive contamination of the Pine River, this site itself, and the surrounding floodplains. DDT within this area, so the Pine River and the floodplains, in some of these sections of this river, reach concentrations over 40,000 parts per million. So to put that in perspective, it takes about 6 parts per billion to kill a mosquito larvae. So almost 1,000-fold higher concentrations are detected in the areas surrounding where this plant used to operate. So extensive contamination within this area of a chemical that was banned from use almost 50 years ago. So it's still there today in extremely high concentrations. So due to this contamination, the Michigan EPA has targeted this site for remediation, and specifically they're looking at using activated carbon as a means of cleaning up the DDT within this area. So activated carbon comes in many shapes and sizes and is used in a lot of different applications. In the context of remediating organic compounds, activated carbon is an extremely effective tool because it essentially acts as a sponge, soaking up organic contaminants from the environment. So if we were to take a look at an example here, so we have some soil that's contaminated with DDT in this habitat and some earthworms living in this area. This DDT that's present in this soil still has its insecticidal properties and could very easily make its way into the tissues of these worms and other invertebrates, affecting the ecosystem within this habitat. Introducing activated carbon into this system as a remediation strategy can help reduce what we call bioavailable DDT concentrations by essentially acting as a sponge and pulling up that DDT out of the soil and holding onto it really tightly. So we're not actually removing DDT from the site, we're just removing or reducing its availability to organisms within this habitat. Reducing that available DDT can help restore ecological integrity of this habitat by kind of reducing that stressor, removing that stressor from that habitat. So with this in mind, the EPA is currently conducting a pilot study looking at the effectiveness of activated carbon as a remediation strategy in the floodplain soils surrounding the Pine River of this Velsical Superfund site. Our objectives as part of our research were to evaluate the effectiveness of activated carbon as a remediation strategy for DDT and its metabolites, which I'll refer to as DDX from here on out, and compare strategies of in-situ remediation to laboratory studies. So a lot of times when remediation with activated carbon is done, they'll oftentimes do a kind of a pilot study by taking some of the sediment or soil back to the lab, adding activated carbon, and seeing how it reduces concentrations under laboratory conditions. They assume that those laboratory conditions will mimic what's happening in situ. But we wanted to kind of see, we had an opportunity to do a side-by-side comparison and see if that's actually true. Do laboratory studies of activated carbon remediation actually predict what's happening in the field? So to do this, this is our study site here. So this is the Pine River, the Superfund site, the actual plant site somewhere up here. This section is the portion of the floodplain soils that the EPA is targeted for their pilot study. And within this site, we focused on 4 of these grids in particular. So we took soil from each of these 4 grids and used them within our study. So to actually compare or look at the effectiveness of activated carbon remediation, we had a couple different treatments set up. So what was done is in the field, activated carbon was applied to the soil's surface by hand spreaders at a rate of about 2% by weight. After that activated carbon was applied to the soil, we went out and collected earthworms that were residing in the soils at 0, 3, and 9 months post-amendment. Those earthworms were then taken back to the lab and we extracted the DDX from their tissues to look at bioaccumulation in the field. Also, at those time points, we went out and collected soil from each of our plots, returned it back to the lab, and conducted laboratory bioassays with worms to see how bioaccumulation was changing under more controlled conditions at these same time points. We also wanted to see how in-situ amendment compared to laboratory amendment. So to do this, before the activated carbon was applied to the field soils, we went out, got jars of dirt from each of our plots, brought them back to the lab, and amended them with the same 2% activated carbon. At each of our time points, we then conducted laboratory bioassays with earthworms at 0, 3, and 9 months to look at how bioaccumulation of DDX was changing in the earthworms over time under laboratory conditions. So to orient you, so this is some of the data that we have here. So on the y-axis, we have DDX concentration in the earthworms, so the actual tissue concentrations of the earthworms. We have our time on the x-axis here, so the 0, 3, and 9 months that we did our sampling. And we have our various treatments organized within each of these time points. So the LA stands for the laboratory amended soil, so that's soil that we collected from the field and amended in the lab. Field, or FA, stands for the field amended soils, so the soil that we collected and brought back to the lab for laboratory bioaccumulation assays. And FC stands for our actual field collected worms, so the worms that we collected from the field that were actually living in these soils. So I'm only going to show you data from one of our 4 plots, but the trends are pretty much the same across all of those trends, and we're seeing total DDX concentration represented within these bars. So within our field collected worms, we saw a general trend of decreasing DDX concentrations within the tissue. So after our first sampling point, we saw a pretty significant reduction in bioaccumulated DDX within the worms, and that trend held true for our 9-month sampling. So it looks like the activated carbon is reducing the bioavailable DDX in the field. This was true as well for our laboratory amended soils as well. So the bioaccumulation assays that we conducted in the lab, we again see a reduction in DDX concentrations within the worms, dropping pretty significantly throughout all of our time points here. So pretty high to begin with, right at the amendment point, and then 3 months and 9 months later, we're seeing the activated carbon is sucking up that DDT and making it less bioavailable. Again, a similar trend in the field amended soils here, but the real interesting thing, or the observation that we made from these data, was that although the field and the lab amended soils were seeing reductions in total DDX bioaccumulation by the earthworms, the extent of that remediation between lab and field was quite different. So we're seeing a much larger drop in bioaccumulated DDX in our lab amended treatment compared to our field amended soils. And we also saw changes in the rate at which this decline in bioavailable concentrations of DDX was occurring. So that is what is represented here. So again, we have DDX concentration on the Y, time on our x-axis, and these lines represent kind of the general trend in decline of average DDX concentration within our earthworms for one of our plots here. So we can see in the blue here that the overall extent of decline in bioaccumulated DDX was much higher for our lab amended soils compared to the field amended soils, and the rate of decline here was significantly faster for the lab amended soils compared to the field. So that's a lot of data to throw at you in a short amount of time. So what does it all mean? Well, the good news is that activated carbon remediation is working. It's reducing bioavailable DDX within our field site. So it's making these soils more tolerant for the earthworms to survive and live in these habitats. But it seems that our laboratory experimentation is over-predicting the time frame over which remediation is occurring. So it seems that under laboratory conditions, the DDX in the soil is being reduced, so the bioavailable concentrations are being reduced much faster than what we're seeing happening in the actual field. One of our hypotheses as to why that might be the case could be differences in condition, environmental condition. So much like Erie, Michigan gets a lot of snow and cold temperatures in the winter, probably not quite as much snow as we get here, but those reduced temperatures might cause differences in the way that we see bioavailable DDX changing between the two treatments here. So those colder temperatures that we're seeing in the field compared to our lab soils that were just held at constant room temperature over those 9 months might be slowing the rate at which the DDX is moving from the soil it's been bound to for 50 years to that activated carbon. So in order to actually see a reduction in bioavailable DDX, we have to see it move from the native soil to the activated carbon. That rate of movement is temperature dependent, such that most of the remediation time that we did this study over, we did our sampling between August and March, occurred over the winter. So that reduced temperature could be slowing that rate of transfer, causing these discrepancies between the lab and the field. Another hypothesis that we had to kind of explain this is related to the way in which the activated carbon was incorporated into the soils. So in the lab, we collected dirt, brought it back, dumped activated carbon in, and then stirred it into that jar. In the field, we just kind of spread it by hand over the surface and let bioturbation and natural processes incorporate it into the soil. So you can see it's kind of blurry in this picture here, but you can see there is a layer of activated carbon that's been incorporated down into the soil here. We assume through bioturbation by the natural earthworms or the worms living in these systems. But the difference in those mixing processes could also account for the differences in remediation rate that we're seeing between the lab and the field. So because we mixed it really good in the lab, we're getting better distribution through the soil, which could help to speed the extent to which that DDX is kind of being absorbed by that activated carbon, while out in the field we're waiting for natural processes to incorporate into that soil, which might slow the rate of reduction within this habitat. So ultimately, in the context of whether or not lab studies can predict field amendments, we kind of came to the conclusion that if you are going to use laboratory studies to predict in-situ remediation, you should do your best to mimic the environmental conditions under which your remediation is occurring. So try and match those two things as closely as possible to get the best prediction of what might be happening. So with that, I'd like to thank Scott, Tom, Frank, and Tom for collecting soils, Dr. Lynn Beatty over at Barron for helping us with the statistical analysis, and then some students up at Alma who did the earthworm bioaccumulation assays, Mia, Nicole, Lillian, and Becca. And with that, I would be happy to answer any questions.
Sam, maybe I missed it, but what was you describe the laboratory control that?
Yeah, so in terms of the remediation?
So you artificially amended some laboratory, and then you had different time series. Did you have a condition where there was unamended soils, and you looked at the change in DDX concentrations?
Total DDX concentrations.
Yeah.
We did some passive sampling with this as well. So SPEME fibers were added to the soils to look at DDX concentrations over time. We didn't have a pure unamended soil that we looked at, but we do have data for like the field concentrations. They did total extractable DDX from the field, and they did see that the trend in DDX concentrations was similar to what we were seeing in the worms. So in the places where the soil was amended, we did see drops in bioavailable DDX, but the overall total extractable concentration, which should have pulled everything, even that which is bound to the activated carbon, remained fairly consistent. So because the activated carbon's not actually removing anything, it's just sequestering it away from organisms, we wouldn't expect a change in total necessarily over time.
Would there be any kind of a metabolism of the DEX in the earth forms over time in the absence of the soil amendment? I mean, how would you account for that?
I mean, you could. There could be that they're taking it up and then breaking it down into some form that's then being metabolized and released. I think that given the time scale of this contamination and the time scale of our study, it's unlikely that it would cause huge changes.
Got a question. So you said that this is at a Superfund site and that this was like a test plot area for treatment. So that test plot was just over these 9 months, right? And then they're going to expand that over a greater area of the Superfund site?
Yeah, so that floodplain, entire floodplain area, they're going to apply activated carbon to that entire region as a kind of remediation of that area.
Okay, and what's the time frame on that, do you think?
That's a good question. I think that they're starting to move on it now or early next year.
Okay, I was just curious.
Yeah.
Do you have any other questions?
I have one question. Is the changes in the Ddt concentrations in the earthworms generational, or is that a change in the like the same earthworm? I don't know how long.
Yeah, the bioaccumulation assays are 28 days.
Okay.
So we put, unexposed worms in, leave them for 28 days, and then pull them out. So it should be the same worms.
Okay.
Any other questions? All right, well, thank you very much.
Thanks.
All right, and up next we have a bit of a slight change to the schedule that we had before, and we're going to be having Sean Dalton present for us on his aerial drone surveys of shorelines at Presque. That is instead of the other 2 presentations that you see listed in the schedule of talks for today. So this will be the last one for today.
Oops.
What's that?
Very good.
They, make no promises.
Do you need help?
It's blue. Why is it blue?
Did you switch the input?
I don't know. Maybe. I don't know. I did something.
Yeah, I was gonna say, what'd you click?
I broke it.
Hey.
I just nudged it too hard. All right. Hello, everyone. As Sarah said, my name is Sean Dalton. I'm the lab and field coordinator here at the Regional Science Consortium, as well as the GIS specialist. Today I'm going to talk to you about a project that we've been doing over the past couple of years, which has been using a drone in order to survey both Presque shoreline as well as some of the primary wetland habitats that have been part of another ongoing restoration project over the past decade. So before I get into the actual drone and the surveys themselves, I am going to give you a brief natural history of western Pennsylvania, just because I know not everybody is familiar with the area, and I'll give you some context as to why we're even looking at what we're looking at. So this whole area, all of the topography and a lot of the geologic formations that you'll see in this region were predominantly formed by the last ice age, which went through this area approximately 23,000 years ago. So the southern lobe of this glacier, which was called the Laurentide Ice Sheet, carved out the Great Lakes. Specifically, the lobe that carved out Lake Erie was called the Lake Erie Lobe. So as you can imagine, when you have a massive wall of ice pushing across a continent, it picks up a whole bunch of debris, sediment, rocks, sticks, stones, and when that glacier melts, it just drops everything right where it was holding it, kind of unceremoniously. So the ice sheet itself, you can see here, is the progression of it as it retreated, as it melted. So you can see at its extent, where's the laser on this? It came all the way down here, just south of us a little bit in this region of Pennsylvania. There's a state park just south of here near Mercer called Moraine State Park. That was the southern boundary in this region. The reason it's named Moraine State Park is that the moraine is the name of the ridge of sediment that's pushed up at the forward edge of the glacier. So that state park is actually on that ancient moraine of this glacier. Another cool thing that I like to point out around here is this area here in Canada, Hudson's Bay, is the entire perimeter of it is a large formation of granite known as the Canadian Shield. There is no granite that naturally occurs in Pennsylvania due to our formations here. We're dominated by a sedimentary bedrock, a lot of sandstone and things like that. However, if you've ever been driving around in some of the glacial valleys in the area, sometimes in the woods or on tops of hills, you'll find these giant boulders of granite. These are what are called Glacial Erratics, and it's quite literally a piece of the Canadian Shield that was picked up by this glacier, dragged all the way here, and when it melted, it was just dropped wherever it was sitting. Because of this, it brings us to Presque Isle. So Presque Isle itself is composed entirely of unconsolidated sediment. There's no geologic formation under the lake that is holding our sediment here. It is quite literally a loose sand pile that has been pushed around the lake basin since it was dropped there by the glacier. So it was dropped in this edge of this escarpment that would fill as the glacier melted to become Lake Erie, and was gradually pushed all around until we have what we have now as Presque Isle. It's hypothesized that the original deposition area of this ridge currently sits about 30 to 50 feet below what the current lake level is. So this is an old figure from the U.S. Army Corps of Engineers that just kind of shows the hypothesized migration of the peninsula throughout time. So if you see in this last figure, this is how it looked in 1930, and you can see this dotted outline is where it was predicted that the peninsula would have been sitting in the lake about 1200 AD. So the neck where this is about where Walnut Creek's outlet is now currently. So in that time, it's gradually migrated down the shoreline towards the east. The reason for this is because in Lake Erie, there's a predominant current which is known as Longshore Drift or a longshore current. We get a lot of our wind that comes out of the northwest here, and as the wind blows across the lake, the waves follow the wind. So as these waves approach the shoreline, it's generally at an angle since we're not getting a straight-on breeze. So as the wave washes up onto shore at an angle, it is going to fall straight back perpendicular to the shoreline because of gravity. So you can imagine if you were a little piece of sand here, you'd be pushed up on the beach, you'd fall back down, you'd be pushed up again, and you'd gradually migrate down the shoreline. So that's the reason as to why the peninsula is doing this. Now, so that brings us to the drone. So Presque Isle currently gets over 4 million visitors per year. A lot of those people like to come and go to the beaches. It's a huge recreational thing every summer, and it brings a lot of money into the local economy. Sand replenishment is a huge project that happens every year led by the DCNR to preserve these beaches and the recreational areas, as well as the habitats that exist here on the peninsula for other species. So because of this, we got this drone. It is called a Dji Phantom 4 Pro V2. This is one of the most common consumer drones that you'll find on the market today. It comes out of the box fitted with a 60 feet per second 4K camera. It's got really nice high definition footage. But we also procured a secondary mount for it, on which we put a secondary Survey 3 camera, which is manufactured by a separate company, and it is in orange, cyan, and near infrared wavelengths. This allows us to look at the reflectance off of plants and assess vegetation health within some of these wetland habitats, which I'll talk about a little bit later. So that being said, there's a lot of stipulations and testing that must happen in order for you to, 1, operate a drone on Pennsylvania State Park lands, and 2, operate a drone for anything outside of recreation where you're being compensated for it in a professional manner. That requires you to go and take an 8-hour in-person course. I took it over at North Coast Air over at the airport. You go through that, and then you have a 2-week period to take a practice test, and then you have to go to an official designated FAA testing center and take the official Part 107 test. Now, my original testing date was scheduled for March 18th of 2020, and if that date sticks out in anybody's mind, it is because it was the day that everything in Pennsylvania shut down because of COVID. So the morning of my test, I got an email that I had been refunded, and that began a long period of waiting as to when I would actually be able to take that test. Eventually, I believe it was July 26th that I was able to take it. So a little bit longer of a period of time, but I managed to retain enough information to pass. So these are showing some of our targets. This is what I've been able to accomplish in the past year and a half, approximately. It's kind of hard to see, but you can see up here along the shoreline, there's kind of that imagery that's it looks different. That's all of the drone imagery that's put on. So we've been able to make it from the edge of Gull Point down to this region here, which is right around near the lighthouse, as well as we've completed surveys of this wetland area out here in between Beach 10 and Beach 11, known as Thompson's Bay, Thompson's Circle wetland area. So there's two different projects that I mentioned that we're using the drone to look at. The one is the shoreline and erosion, and the second is the wetland habitats. So the shoreline and erosion I already mentioned is important because of all the recreation and things that we have going on at the peninsula. This was a photo that I went out and I took on April of 2020. We had a storm event, and this is right at the Beach 7 waterworks area where there's that nice walkway that they have. That entire beach had been eroded away, and it was actually, that walkway was collapsing into the lake at that point. So that was pretty impressive that the lake was able to move that much sediment that quickly from one event. If you go back here now today, the sand is almost all the way out until here. So it's pretty impressive how quickly the shoreline can actually change here. Another issue that we have here are these structures called tombolos, which can form. It's kind of hard to see in this picture, but the breakwaters that are offshore of the peninsula, as waves come into them and their energy is dissipated, on the shore side of that breakwall, where the energy of the water becomes low, sand will begin to fall out of that water column just due to the low energy. And it will begin to form these long structures that reach out towards the center of the actual breakwall itself. If these connect to the breakwall, it will actually cut off that upshore sediment source from that drift, and you'll start to get a lot of erosion on that down current side of the beach, which I'll show you a picture of. But then the other zone are the primary wetland habitats. The reason we're looking at these is this is part of another project in which we're going into primary wetland habitats that have been overrun with a lot of invasive aquatic plants. So particularly on Presque Isle, there are two big invasive plants in these wetlands. There's the narrow-leafed cattail as well as Phragmites. Phragmites is a common reed, and narrow-leaf cattail is a different variety of cattail to the native cattail that we have on the peninsula. And you might be thinking, if it's a non-native cattail, what's the difference between that and an invasive cattail? Well, the invasive cattails actually grow so closely to one another that fish and amphibians and other sorts of animals that would use that area for habitat normally physically cannot get in between the stalks and have adequate space for them to reproduce and do other things that they would do in this habitat. So it essentially chokes them all out, and except for things, I believe red-winged blackbirds are one of the only things that are able to utilize that area once it becomes that dense. Previously, when I started back around 2014 or so, this was freshly sprayed down and was kind of looked like a big mud pit. Before that, it had been 10, 11-foot tall Phragmites across the whole thing. So this is what I was talking about. This is around the Beach 9 area. So you can see here this tombolo has formed. This is my drone imagery that I collected in 2020 compared to a 2012 satellite image. So you can see as this tombolo forms and connects here, you can see all of this sand that's eroded downstream because there's no longer this source of sediment pushing down the shoreline. So this is another area out on Gull Point that I wanted to point out. Just in 2016, this was an area actually that we had been hiking out to and monitoring. Right at this area, it previously breached, and the lake was actually flowing directly into Thompson's Bay. For a brief moment of time, Gull Point was Gull Island. Now it has built back up a little bit, but if you notice here, this is where the previous shoreline was. So you can see how far back it's fallen. I've measured it, that's about 70 feet directly south that the shoreline's fallen. Now that being said, that sediment is always being pushed down the shoreline, and if you notice here, Gull Point has extended further out now. So although I haven't gotten a full survey of the entire Gull Point area, just because it's so large and we're limited by battery and things like that, I think there's probably considerably more sediment here. However, it does look like quite a bit has been lost, and we've been pushed farther south on the shoreline. So this is the Thompson Circle wetland area. This is just the visible light image of it. Is not the near infrared or anything. I'll show you that next. But just to see the difference between in 2012, when everything was sprayed, just how much of that vegetation has actually begun to come back now. So now that brings me to the Ndvi and the OCN sensor. So an Ndvi is a normalized difference vegetation index. So essentially what this is a graphical indicator that you can use to assess plant health. So due to photosynthesis, plants strongly absorb light in the visible light spectrum, and they reflect a high amount of near-infrared light. If a plant becomes stressed at all, it may look identical to your eye, but if you look at them within these wavelengths, they show up totally different. So the way this camera works is it takes these photos in it and you run it through a software that then codes every pixel with a value from 0 to 255, which corresponds to an already established formula for this Ndvi. So after you go through and you assign all of those values to those pixels, within those ranges you can apply a color ramp to that and you get a nice red to green variation of where your reds and yellows are things like bare soil or water, and then your yellows into greens are different ranges of vegetation health, density, and things like that. Traditionally, this is performed with a red-green and near-infrared sensor. However, this is kind of a proprietary sensor that we purchased for this with orange, cyan, and near-infrared. And the reason for that is that, so this is the normal sensor's wavelength. By extending that wavelength range just slightly, you can see you get much more contrast and definition into those areas. There's less noise in areas where you have bare soil, and you just get a much clearer picture. So we were able this past summer to do a flight of that Thompson's Bay wetland area. And if it loads, there it is. So you can see here, here's the visible light next to that Ndvi that we have. So in here, all of this red and extra noise, that's the water. It's reflecting some sediment and things that's in suspension there. But you can see in here where the vegetation is much denser, you get those deeper greens and things like that. So with the real, I guess, utilization that you can do of this is now, if we were to do successive flights, you can then compare season to season vegetative health, as well as you can do multiple flights during a season and see how the health of that situation is changing over time. So in places like this where they're going in and they're spraying and treating with herbicides, if we know there's a stand of invasive plants that we need to hit here, you can spray, do a flyover following week, and see the efficacy, your overspray, and how well you actually targeted and hit those areas. As well as then looking at it season to season, you can see how the plant health may be increasing or decreasing based on what the inputs into the system may have been. So the image processing itself is very simple. The drone operates through an app that runs off of my iPhone. It's pretty easy. It's just a GPS. You can click and draw a bunch of boxes, and then it pre-programs a flight, and you click fly, and it takes off, and it goes. There's a micro SD card on the drone itself, which when you're done, you pull it off. There's a website that's coupled with the app. You make a username, password, log your images in there, and it automatically mosaics it for you. It's really fast as well. There have been days where we've been out, we've done a survey at 8 a.m., had it in the computer by 10, and then before 4 o'clock that day, we have it in the computer what that survey looked like that same day. That being said, there are some difficulties that come along with this, with how user-friendly it is. Weather can be very unpredictable. Whenever you're flying, you always want good, calm, clear weather. One, for the image, and two, it's bad if the drone crashes. You're unable to operate over people unaffiliated with the mission, which can be very challenging when you're trying to survey public beaches that have thousands of visitors every summer. So usually when we're doing those beach surveys, we have to get out there very early in the morning before any park patrons are on the beaches, because DCNR doesn't like when we kick people off the beaches to do drone surveys. Battery time is also another limiting factor. Each battery only lasts about 30 minutes. We have 3 of them, so we're able to fly for about an hour and a half, but that changes based on temperature. As things get colder, your battery time shortens. Wind speed is also another issue. If the wind speed ever exceeds 1/3 of the maximum velocity of the drone for any given survey, you get a high wind warning and it kind of shuts itself down. It doesn't really work out, which can be very challenging when you're surveying a shoreline because gusts will kick up every now and then, and sometimes it seems fine and it gets up there and 150 feet above the shoreline when you think it's calm, it's actually pretty windy. Controlled airspace is another big one. So the type of airport that Erie is, from their control tower, there's a 5-mile radius around it in which you have to have special permission to fly, as well as there's set ceilings on which you can operate. So that airspace extends over the peninsula until about the waterworks ponds area. You can fly on that part of the peninsula, but your maximum altitude is 50 feet. So if you're trying to do a survey of a wooded area where there's trees and things like that, it's not really great because then your drone's going to fly itself right into a tree and then you're not going to get any data from it. Time of year is also difficult. I also manage the summer interns that come here to collect beach samples and things for the park. So it's not always ideal timing. Some days I can't get out there and fly when I need to, and leaf off, leaf on. There's very precise times of year in which you really want to get these surveys done so that you're getting a good representative idea of what that vegetation's health actually is. And sometimes you miss it with other things going on around there. And COVID-19, the ever-evolving pandemic of inconvenience. It was, we made it through, but it's a little difficult when you're waiting 2 weeks to take a test and then you have no idea when it's going to come and you can't go into the testing center and study anymore. So our future plans and capabilities now, we're going to continue building this shoreline database just because the shoreline changes so rapidly every year. We're able to then compare that with the ongoing database that we have of the RSC buoy fleet that is in the lake every summer. We're going to continue doing more Ndvi analyses of the Ducks Unlimited habitat restoration areas to get those month-to-month comparisons as well as those season-to-season comparisons as well to continue to monitor how this wetland restoration project is evolving. As well as we have the capability of looking at new sample sites that you wouldn't be able to access normally or monitor, and just the ability to have aerial photography of any one of your sample sites that day is extremely powerful, particularly when you're talking about an environment as dynamic as the beaches and shoreline of Presque. Because it's not just a season-to-season change, it can be storm event to storm event. You can go out tomorrow if there's a storm tonight, it could look totally different. So being able to get that spatial data that quickly is a very powerful tool. So with that, I'd like to acknowledge some of the project partners that have worked with this on us: Ducks Unlimited, PADC and ARCO, Native Erie, California University of Pennsylvania, and the Erie Bird Observatory. And with that, I will take any questions. Yeah.
Is your goal with herbicide work, is that open marsh, or do you want the replacement of the narrow leaves?
So it's a so the idea is to remove the invasives, propagate native plant species that would be there, and now we're monitoring both the plant health as well as another part of that project is monitoring amphibian and fish comeback. So it's kind of another piece of this greater puzzle of just trying to get these primary wetland habitats back to their more natural state with the invasives. Yes. Yep. Yep. Jen Salem has actually already begun doing that.
Do you try to do your Ndvi analysis or sampling, I guess, the same time each year so that there's not seasonal changes in them?
Yeah, so ideally with the Ndvi, the second week of July is the best time to do it because that's like peak vegetation health. That being said, it's still good to get something earlier and later in the season just so you can see that change over time. But yeah, ideally, if you could do July 12th, 2021, July 12th, 2022, that'd be ideal. But like I said, with timing and scheduling, that doesn't always happen.
I have a question. So, with regular satellite imagery like that, everything was compared to, is there a regular schedule on which that is updated at any point, or is it kind of random?
I'm not really sure.
Okay, so I work with you on the project, but it's like, I know we have different satellite images, like one of them was 2012, one of them was 2014, and that kind of different stuff.
so there's always a new satellite imagery that's available. It's not always free, which is the issue. So Pennsylvania has what the PASDA, Pennsylvania Spatial Data Database, where you can go online and all of the free public use data for Pennsylvania, you can download it. That's the most up-to-date Landsat imagery for this area currently, which was actually Sean Rafferty from Sea Grant was the one that had that, paid for that, and had that flyover done and made it public access. So thanks, Sean.
Sean, really cool stuff. Is there any application to submerged aquatic vegetation in the sea through the water?
You know, I'm not really sure. I haven't looked into it specifically. I will say that it's kind of hard because when you get to altitude, even some of the plants that are on land, you can't necessarily make out what they are. Some of the places in the bay too, there's a lot of sediment, so you don't really see past the top layer. That being said, we have done flights over Thompson's Bay where you can see where stands start and approximately the area that it covers if the water's fairly clear. But I'm not really sure how effective it would actually be for those applications. But I mean, if you ever want to go out and try it out.
All right, good. And